Hi everyone, my name is Maya Sprouse, and today I'm going to be giving a very rough draft of my talk on sperm ribosomal DNA copy number in our attempt to distinguish it as a biomarker for male factor infertility. A quick overview of the talk: I'm going to outline the societal impact of fertility studies and why research specifically in male fertility is an area of interest. I'll provide some background info on ribosomes and why we chose to study the multi-copy genes that encode them. Then I'll outline the methodology and the key findings of our study and suggest some areas of future research given the things that we have discovered. So for now, you guys can kind of ignore this image. I just wanted to throw some kind of figure on the slide for now. But the World Health Organization defines clinical infertility as the inability for sexually active non-contraceptive using couples to achieve pregnancy in the span of 1 year. This disorder affects about 15% of couples nationwide, and the male factor infertility donates to about 40 to 50% of these cases. Now, male infertility is a multifaceted disorder, and so contributors are broad, and they can be reflected by poor semen parameters, abnormalities in endocrine function, as well as atypical anatomical structures related to gamete production. Despite the broad impact of infertility, the complex nature of fertility makes understanding the contributing mechanisms very difficult. In fact, in 30 to 40% of cases, the underlying causes of male factor infertility are indiscernible. It's for this reason that in the past few years, research has really focused on identifying biomarkers and mechanisms related to male fertility. This brings us to our objective. We are aiming to explore one potential biomarker in ribosomal DNA copy number in hopes of identifying a diagnostic tool for male fertility, which would enable the better quantification of the disorder. So a bit about ribosomes. This organelle is the site of all protein biosynthesis in the cell. They're made up of a large and a small subunit, and the constituent parts of each of these subunits is located on different loci on different chromosomes. The 5S subunit is part of that larger subunit in the ribosomes, and it's located only on chromosome 1. Contrastingly, the 5.8S, the 28S, and the 18S are all part of the 45S subunit, and they are found at a completely different locus located on chromosomes 13, 14, 15, 21, and 22. Now, despite protein synthesis being a crucial part of cellular functions such as metabolism, growth, damage repair, and transport, the number of genes encoding for these ribosomal components, or the ribosomal copy number, varies not only between species, but also between individuals within the same species. The 5S and the 45S subunits are genetically unlinked, but a phenomenon has been observed where the 45S and the 5S copy number is concerted, usually existing in a specific ratio within individuals. There's also data suggesting that variation from this concerted ratio is associated with disease states. So I don't have access to any of these figures yet, which I apologize for, but at this point in the presentation, I'm going to explain a bit of the work done previously by the Environmental Epigenetics Lab at UMass. Last year, one of the researchers identified mitochondrial DNA copy number and deletions as strong diagnostic tools for male fertility. Additionally, a recent study out of China showed that ribosomal copy number and mitochondrial copy number fluctuated together within gastric tumor cells. So taken together, these two findings suggest that ribosomal copy number may be indicative of a disease state, namely male infertility. So again, I don't quite have all the specifics here yet, but I'm working to track them down. A bit about our methodology, though. We're working with a sample size of 180 individuals from the SEEDS cohort. SEEDS stands for Sperm Environmental Epigenetics Development Study. These individuals are all enrolled in the Bay State Medical Center IVF clinic. And then I will go into a bit more detail about how the samples were collected, how the DNA was isolated, and how the semen parameters were determined using the World Health Organization standards. So here are the specific semen parameters that we are looking at. Concentration, count, motility, and morphology are measured and assessed by the World Health Organization evaluation for normal versus infertile standards. If an individual sample has low parameters, they are essentially more infertile. So at this point, I'm going to start getting into detail about the data that we are currently in the middle of collecting. We're using a triplex qPCR to assess the copy number and are using a single copy p53 gene for copy number determination and analysis. Essentially, the assessment of the concerted copy number between the 5S and the 18S subunits versus semen parameters will allow us to determine if our DNA copy is a predictive tool for fertility. If we find that it is a predictive tool for fertility, I will suggest future research in the areas of epidemiological studies and try to determine some factors that may play into this copy number variation that we see between individuals and generations. I would then want to acknowledge the people in the lab that have helped in this research study: Dr. Rick Pilsner, the PI, Emily Houle, Amos Fidel, Noah Don, and Becky Wolf. And then finally move on to contact information for anyone that has any comments or questions. Thank you all for listening, and let me know if you have any feedback for me. Thank you.